All right, good morning, everyone. Welcome to TeleShadowing. We are live on YouTube. Feel free to ask questions in the chat box as we go along, and they will be addressed. Today, we're going to be discussing vaccines, immunity, basics on sort of how our immune systems actually work too. And I would say this is a really important topic as we start off our infectious diseases month, primarily because this is stuff that's taught during the first semester of medical school. And I'm in my first year currently of medical school, and I wish I had a head start like we're going to be giving you guys today, so that it won't be so overwhelming when you do learn it when you do start your medical school journey. And so today, this discussion will actually be led by Hamza Ahmed, who is a current BSDO dual degree student at the Seton Hill University and the Lake Erie College of Osteopathic Medicine. So, Hamza, feel free to take it away, and I'm excited to learn.
Sure, thank you. Yeah, let's begin right now. So, before we actually learn about how vaccines work and how our immune system works, one of the most important things we should look into is the history of vaccine, how they developed, how immunization developed, because that'll give us more appreciation for the science behind everything. So, this is the outline. We'll be beginning with vaccine history. Then we'll talk about the human immune system. Vaccines, we'll just learn about how they work. And then finally, we'll discuss vaccine hesitancy because that's a very, very relevant topic. And I think anybody who's going into the medical field should be able to learn about it and learn how to deal with it. So again, before we actually begin, remember to understand differences. People genuinely do have a lack of trust due to genuine past grievances and historical marginalization. People aren't just against the vaccine because they feel like it. Nobody wants to make their life harder just because, I mean, maybe there's are some people who do, but the overwhelming majority of people genuinely do have concerns and they genuinely should be addressed. Don't be arrogant towards these people. I know a lot of people in the not necessarily medical field, but a lot of people who are getting a higher education, let's say it's college or whatever, tend to have a bit of a superiority complex when they're approaching people who have views that are mistaken. Again, these people are not lower than you for having these views. These people genuinely do have concerns. Finally, we should ask questions ourselves rather than blind following. So, for example, the reason that people are concerned about the CDC or about the COVID vaccine is because they're asking questions all the time. We, on the other hand, should not just listen blindly to what the CDC or whatever everyone is saying either. Why? Because if somebody's asking us questions about, why is the vaccine doing this? Why is the CDC doing this? The only answer we have is, oh, well, this government organization you don't trust must be right. You have to actually be able to explain the reasoning behind why they are right, because otherwise nobody's really going to listen to you. You need to build the credibility of the organization. The organizations do not have credibility anymore. And as sad as that is, that has genuine background. And we'll go into more of that near the very end. So vaccine history, let's get some historical context in here. So a pre-vaccine world is essentially a world without microbiology. People thought that bad air or miasms caused the disease, and this is actually a concept that still exists in homeopathic medicine. Homeopathic medicine is like a very small subgroup, and I think they reject a lot of widely accepted medical practices, medical theories. Now, actually, if you look at the right here, the reason I chose this picture, this is a artistic depiction of the Black Plague. Nowadays, we know that Black Plague was called by the bubonic plague, and something interesting is that the bubonic plague is popping up, every couple of years. Like I think it was just in China a couple months ago, or like a year or two ago. But we're not like terrified of it anymore. It's a disease. We understand that it's caused by this disease. We know how to deal with it. We know to wash our hands. We know how to sanitize our surroundings. We know we have all these treatments for it, but at the time, it was just some force that just existed. People believe that it's a result, again, of bad air, or it was the result of their wells being poisoned. Nobody actually knew what it was. It was just this giant event that occurred out of the blue, and that's how people viewed it at the time. Now, concepts like quarantine still existed though at the time. Like for example, Ibn Sina, he was a Persian philosopher, physician, many things that he did. But he was somebody who argued for quarantine back in 1025. This is, was it 2024 now? So just a little over, a little less than 1,000 years ago, he argued for quarantine. Um, so concepts like that still existed. There was still some idea of disease being infectious, but it wasn't truly what we have now. At the time, it was mainly just bad air or poison that caused disease, not physical pathogens. Now, microbiology, as I said, we were in a pre-microbiology world. Robert Hooke is somebody who discovered the cell. So Robert Hooke here is, I believe he was a scientist. I actually should have included this, but he built this microscope here we see right here. And he was the first individual to actually look at a cell. And he named it a cell just because of the way it looked, right? Everything was separated from each other. So Robert Hooke is the first individual to discover a cell. He discovered that this is what makes up all life, and a lot of our understanding of cells started from here. So this is like the first step in understanding cells. We still didn't know about pathogens. We still didn't know about diseases. We still didn't even know about bacteria yet, right? That came much later. So this is just a first step. At this point, we still think miasms and poison cause the disease. Now, Antony van Leeuwenhoek, this was a Dutch person. He also created a microscope. So actually, like if you see right here, this is a microscope that he made, a little diagram of it. And he was the first one to actually identify microbes. So actually, if you look over here, this little diagram on the top right, this is actually these are all drawings of the things he observed. So this looks like a mosquito larva, actually. I think these are all mosquito larvae in different phases of their life, unless I'm mistaken. Um, but the most important thing here is he took a bit of dental plaque from his teeth, obviously, and he observed it under a microscope. This isn't exactly a picture of what he saw. We don't really have photos from back then, but this little line right here, that's bacteria. So Antony von Leeuwenhoek, he was the first person to actually observe bacteria to begin with. Now, Louis Pasteur, we're going to be hearing his name a lot. And actually, you guys probably have heard his name a lot as well without realizing it, assuming, you haven't taken microbiology already. But Louis Pasteur was also very important because he's the one who disproved the concept of spontaneous generation. Once we had this idea of bacteria, um, people still believe that life spontaneously generated, bacteria spontaneously generated. I think even Darwin, I may be wrong about this claim. I'll have to look into it. So don't take my word 100%. If somebody can like confirm it and send me in the chat, then let me know. But I believe Darwin even had the belief that frogs literally evolved in mud or came out of mud and spontaneously just were alive. Again, I could be wrong, but we'll confirm. Oh, okay, that's correct. Yeah. So this is generally the belief at the time. But what Louis Pasteur did was he used his famous experiment where he applied heat to this flask here. This flask had this giant tube which would prevent air from entering. So if air was prevented from entering, bacteria would be prevented from entering. So if you apply heat and let the flask sit, if bacteria were to spontaneously generate, there should be bacteria, but there wasn't any. Then he applied heat, removed the neck again, this time, and bacteria was present. And finally, the last situation was the heat was applied, the flask was tilted sideways, and bacteria was present. So this proved that bacteria was not something that just spontaneously appeared, rather it was something that came in from the environment. And this experiment was actually what proved that. Robert Koch. Now, I think this is the last scientist we'll be discovering, but he's actually the one who finally discovered the link between bacteria and disease. So he actually discovered the tuberculosis bacteria and discovered that in the context of disease. He used staining methods that we use to this very day to identify the bacterium, and he believed that this means that the bacteria and the disease are related to one another. And essentially, this was the birth of the concept of infectious disease. Because again, even up to this point, right here, it wasn't confirmed that bacteria is what causes this illness or illnesses. Obviously, not illnesses are caused by bacteria, but this is what confirmed that infectious diseases do exist. And that was the essentially where we begin with vaccines. So this is a quick overview of vaccines. Again, Louis Pasteur shows up here again. So people were intentionally exposed to smallpox from the 1400s to the 1700s, just not in the Western world, not in Europe. Well, I'll explain that a bit more. Now, cowpox was discovered to protect humans from smallpox by Benjamin Jesty. And Dr. Edward Jenner was the first to inoculate cowpox to actually vaccinate. Again, more detail soon. Louis Pasteur develops a lab-made vaccine for cholera and rabies, and then everything we know now continues from there. So vaccines as a process always existed. They weren't called vaccines at the time, but the process that we know as vaccines always existed. For example, breastfeeding mothers pass their antibodies to their children temporarily. So any disease that the mother has antibodies for, the children will have for whatever period of time that they're breastfeeding. Or another example here is that Lady Mary Wortley Montagu, her husband was a diplomat to the Ottoman Empire, and she saw the Turks were purposely inoculating themselves for smallpox for immunity when her husband was a diplomat there. So this is the little diagram here. This is actually Lady Mary Wortley Montagu, but this is a diagram here showing the Ottomans purposely getting my bad, I said smallpox. It should be cowpox. Getting cowpox so that they don't have to worry about smallpox because smallpox is a much deadlier disease. So this is still technically vaccination, even though it's not what we know of today. It's not getting an injection or taking it from the mouth. It's still technically the same role that vaccination plays. Why? Because vaccination is essentially using your natural immune response to make yourself immune to a disease. It's nothing new. Technically speaking, every time you get sick, you're technically getting vaccinated. So if you get the flu and that makes you immune to the flu for a bit, you were technically vaccinated in a way. Now, you didn't purposely get it. You aren't immune to any new strands of the flu that may appear in a couple of years, but you still got the flu, and as a result, your body knows how to fight it. Now, cowpox versus smallpox. Here we have Benjamin Jesty that we mentioned earlier. So cowpox is far less severe than smallpox. Smallpox is a very, very deadly disease, so much so that the World Health Organization made it one of their goals to eradicate smallpox. They're both in the Poxviridae family of viruses, so they are related. If you actually look, so this actually includes chickenpox, cowpox, monkeypox. If you look at the symptoms of these two diseases, they all have the sores that appear on your body, on your skin. And Benjamin Jesty noticed that people who received cowpox would not be affected by smallpox. And he was actually the first person to really notice this. But he never actually published his findings. The publishing of the findings would come from Edward Jenner, Dr. Edward Jenner. So Dr. Edward Jenner purposely exposed milkmaids to cowpox. This is in a farm situation. And so noting their general resistance to the deadlier cowpox treatment was obviously a success. So this became the first real vaccination. And the word vaccination, again, because we're talking about cowpox comes from cows, comes from vaca, or cow in Latin. Anybody who speaks Spanish here also knows that this is the same word in Spanish for cow. So here's an interesting story. Now, Louis Pasteur once again shows up, and for all you know, you're probably going to see him in whatever other studies you do because he's very important in this field. So he contributed to both vaccines and sterilization. So he used attenuated microbes. Again, we'll explain what this is. For now, just say, oh, no, that it means weakened microbes. They're not as strong. They can't really hurt you as much. To fight diseases like fowl cholera. So this is a vaccine is preventional. You get the before you even get the disease, and rabies. So rabies, you actually get that shot after you get infected because rabies works slow. You're able to actually get a vaccine before you even get symptoms. So this became the first lab-made vaccine. Pasteurization. This is when I said that you guys probably know Louis Pasteur without knowing. If you guys get pasteurized milk from the store, that's a process that's named after him. Essentially, heating milk for a long period, or heating any food for a long period of time to kill microbes. So this isn't boiling necessarily. It's not boiling water because boiling water is over 100 degrees Celsius. This is heating it at a little bit below 100 degrees Celsius for a long period of time. Eventually, that kills all the bacteria that is in the product. So that's a process, again, named after him. So he became very important for three things. And there's a lot more things that he's important with. And the funny thing is, he's not even a biologist, he's a chemist. So it's a fun little dip outside of his main job, but still, he made it such a giant contribution to it. So, yeah, this is the first lab-made vaccine. And then I'll cover some vaccine milestones. We had the flu vaccine in 1945. This came after the uh, flu epidemic that came after World War One. This became one of the top priorities of the United States to develop a flu vaccine because it became really bad. I mean, millions of people died during the flu epidemic that happened after World War One, or pandemic, my bad. That's the wrong term to use. The polio vaccine in 1954 was also a huge deal because polio is a very, very bad disease. I don't know if you guys have ever seen the iron lung. I probably should have put a picture in here. But the iron lung was essentially something that they put people with polio in to help them breathe. Um, President Franklin Delano Roosevelt had polio, and as a result, that's why he wasn't able to walk. Mass vaccination came in 1967 for smallpox. Again, this is by the World Health Organization. And this was another huge deal because now you're finally able to give vaccines to the people into the masses. Then polio becomes endemic by 1988, meaning that's only found in certain countries. It's not a worldwide disease anymore. It's found in certain places, and their goal was to, I believe, wipe it out by 2000. And even though they weren't able to wipe it out by 2003, I think it was only found in maybe 16 countries. And then finally, they had a vaccine bank made in 2021. So in the situation of any new disease, any new pandemic, they had the vaccine ready to respond, assuming that this is a disease that already exists. If it's a new disease, then they'll have to develop a new vaccine. So vaccines changed the world. As I said, it almost eradicated polio. It made the flu a lot more manageable. Like just to understand, if you get the flu now, it becomes a bit of a nuisance. You have to take off from work, you have to take off from class, whatever it is. But it's not, a life-threatening situation. You don't always have to go to the hospital. It's possible you may have to go to the hospital, but it's not a requirement that you have to go there to survive. You probably sit in your room for a couple of days, get out. And even then, if you take the flu vaccine, it makes it even better because now your body is fighting it a lot more effectively. And it made many other already existing diseases much more manageable. And infectious diseases, which are already rare due to good sanitation. I think I didn't explain that enough. So I mentioned with Louis Pasteur, with pasteurization, that sanitation helps. Everything in general, we do sanitation much better nowadays. We all wash our hands now. In surgery, we wash our hands. Before we get an injection, they put isopropyl alcohol on us. Everything nowadays is a lot cleaner. And as a result, infectious diseases are also a lot less common. But even those that slip through the cracks, now you have vaccines to help us deal with that. So now we get into a little bit about misinformation. We'll get into it more later, but we'll touch on it a little bit now. So there's two infamous studies by Dr. Andrew Wakefield, and these are two very flawed studies as well. This is something I remember seeing when I was maybe in high school, maybe even before that, talking about vaccines. I think during the Trump presidency, I saw these things pop up a bit because during the Trump presidency, a lot of hesitancy about vaccines became mainstream. So these two studies were both trying to argue that vaccines cause autism. And the first study showed that children developing autism, like showed children developing autism within a month of taking the MMR vaccine. Now they had 12 children, all of which had developmental issues, and eight of those children developed autism. And this study was supposed to be a big deal. I mean, how do you have this many kids, end up being hit so hard by this vaccine? The issue is something that a lot of critics pointed out was, this is in the UK where 90% of children take this vaccine. You don't see 60% of those children developing autism. So that's why this is a very major criticism of this because this makes no sense. How can it be such a high rate here and not a low rate in the real world? So that's one major flaw with this. But regardless, a lot of people do cite this study. Why? Because it's human nature to be cautious. If you see that there's something that may cause a 25% risk to you, or in this case, it's a much higher than 25% risk, you're going to be a lot more cautious. You're not going to be, 1 out of 4 chance that something really destroys your life. You're not going to take that risk at all. But in reality, this is a very, very flawed study that pushes the danger a lot more than it really is. Now, the second study showed the measles vaccine, and they did a intestinal biopsy of a group of children who had autism, who were also dealing with some kind of intestinal pain, so some kind of abdominal pain. And they found that the children actually had measles cells or found in the tissue of their intestines. And again, this becomes very concerning, right? Because you just took the measles vaccine, but now you have straight up measles cells and measles DNA. I think it was DNA actually, not cells found in the tissue. And again, this is going to be causing a lot of concern. A lot of people are going to be concerned about this. But here's the thing. The measles vaccine is live attenuated. You're going to have cells, right? As I mentioned before, the attenuated cell vaccine is literally just weakened pathogen that is put inside you. And the way our bodies work is that we take in the DNA of any diseases that we get, or especially viruses. I think a good chunk of the human genome is actually the result of viruses from the past that ended up getting into our genome because that's how viruses work. They kind of inject their own genetic material into our cells. So the fact that measles cells were found is not too shocking, or even measles DNA. Why? Because this is just how it works. Their DNA is going to be floating inside you because they were literally put inside you. And sadly, even though these studies have been very, very criticized, a lot of people are still very, very concerned and they take these studies as fact. On top of that, a lot of it is fear of the unknown. If you are afraid of the dark, that's not because you're afraid of just darkness or the color black or whatever. You're scared because you can't see your surroundings anymore. That's the same situation with vaccines. People don't really understand what vaccines are. Vaccines are just this thing that's stuck inside you and it does this magic work. Like if you look at this image, actually, this is just clear liquid that they're sticking inside you, and somehow this works. And people are concerned, like, how is this supposed to work? What does this do to me? What is this doing to my body? Why is it a clear liquid? Why don't I see any pathogen? So fear of the unknown is also a major, major role that plays in vaccine hesitancy because if people don't know what they are taking into their bodies, they're going to be a lot more concerned about what is going on. So to reduce the fear of vaccines, we need to stop fearing the unknown and we need to help people stop fearing the unknown by making vaccines something known. So now let's discuss human immunity. This is going to be a fairly long section, and this is going to be one that's a lot more technical than the prior sections. So we have two types of immunity. We have innate immunity and adaptive immunity. And even in innate immunity, you have immediate and induced. So we actually have a little diagram here that kind of explains it. Our innate immunity is, as it says, something innate to us. It's something natural to us. As you guys are looking at me right now, you guys are seeing the skin of my face. The skin of my face is what's preventing any bacteria in the air from getting inside my body, right? So that's one example of innate immunity we actually have right here, physical barriers. Then there's chemical barriers too. So while we're on the topic of skin, I think this actually is oil on purpose. Sebum, the oil which your skin produces, is naturally antibacterial or antimicrobial. It actually kills microbes that get onto your skin. And then finally, cellular defenses. So if something does somehow get through, there are actually some cells, some like white blood cells, that would immediately deal with the disease before it's even really able to do anything to you with minor damage to your body. And finally, there's adaptive immunity. There's active immunity, which is, you get sick or you get vaccinated and you end up uh, having to have an immune response, an inflammatory response, and you deal with the disease that way. Or it's passive immunity. For example, as I mentioned before, the example of a parent breastfeeding her child. So these are the two examples that are given of innate immunity and adaptive immunity. Now, I'll just show you this little diagram here, which actually shows um, how the two types of immunities work and how they relate to one another, actually. So the immediate innate immune response, the pathogen enters and it starts proliferating. It goes everywhere. The pathogen is recognized by preformed effectors. I'll explain what that is. Those are specific cells. And there's two options. Either it's eliminated and the infection completely ends, and there's maybe some minor damage to the tissue. You probably wouldn't even notice it that much. And that's it. Or the pathogen is not eliminated and an induced innate immune response is made. So notice the time difference though, 0 to 4 hours versus 4 hours to 4 days. So now the pathogen invades tissue and proliferates. The activation of cells resident in the infected tissue, so in that specific area occur, and there will be inflammation there, fever, and acute phase response. I'll actually explain how this works too, because it's actually really cool how your inflammatory response works. And again, either um, either the cells are able to attack the pathogen, which means that they're able to kill the pathogen, and then again, moderate tissue damage occurs, or they're not. So hopefully, in most cases, your cells are able to deal with it on their own. They recognize and attack the pathogen on their own. They don't need to do anything else. But if the pathogen is not eliminated, then they need an adaptive immune response. And this is 4 days until however long it takes to kill the pathogen. So the pathogen invades the tissue and it proliferates. The secondary lymphoid tissue close to the infected tissue is made aware of the infection. And the pathogen-specific B cells and T cells, these are white blood cells, are identified in secondary lymphoid tissue. And these B and T cells spread everywhere. And their whole job is to B cells are to make antibodies and T cells, travel to the infected site. And eventually the pathogen should be eliminated and the infection should end. This is going to be hurting a lot more. There's going to be a lot more major tissue damage, but it'll be repaired hopefully. Either that or you die. So those are the two options that your body has when you have adaptive immunity being used. So let's discuss the two types of immunity in a bit more detail. So we have cells that recognize molecules that are prevalent in pathogens. So these are, for example, called pattern recognition receptors. These are on top of cells actually, on white blood cells. And one of those types are toll-like receptors. So pattern recognition receptors, they can recognize molecules that are often found in pathogens. So if there's something like, I want to give the example of the spike protein in COVID, something similar to that. I don't know if the spike protein itself would count, but if they find something similar to that, they would be able to say like, okay, this is probably a pathogen. Let's deal with this. That's actually why, in general, if you have something inside your body that's not supposed to be there, that's why it hurts. So if you're getting a shot and it really hurts when the needle's in there and it stops when it's out, it's not because the needle being in there causes more damage than not being there. It's just the fact that there's some foreign body in there and your body is trying to get rid of it. So pattern recognition receptors and toll-like receptors, which by the way recognize pathogens directly and signal for an inflammatory response in the first place, These are the reasons why our body isn't attacking our own cells, but rather they attack outside bodies. Now, the job of antibodies are produced again by our cells. They opsonize pathogen peptides, which basically is just big words for saying that they bond with certain peptides and antigens, and they just kill them. Antigen presenting cells are white blood cells that take in, like literally take in the um, entire pathogen, and antigens, which are actually parts of the cell or parts of the pathogen, which are recognizable, are presented to, let's say, the other white blood cells. Now, the major histocompatibility complex is something that is used to essentially deal with the cells. So MHC1 is something that's used for viruses, and MHC2 is for bacterial infections. So the T cells destroy the virus. CD4 T cells differentiate and stimulate production of IgG and IgM antibodies. I'm not going to get too much detail into them. They're just different type of antibodies. And then they create memory B cells. So what is the purpose of memory B cells? They're basically there to make sure that in the situation that the same pathogen pops up again, we already have the antibodies to deal with it. Now, this is the adaptive immune response. This is a lot more, in depth than what I had before. But the adaptive immune response includes dendritic cells. So actually, I didn't explain yet. So there are many different types of cells. So there's macrophages, monocytes, natural killer cells. Oh, we actually have a diagram right here too. So macrophages are essentially, it kind of says it in the name, they take in phages. Let me move this out of the way. Here, you actually see them taking a phagosome. A phagosome is like a hole inside of the bacteria or in the cell. Their job is to literally eat up cells. And you have natural killer cells here. Again, kills the virus infected cells. So it actually kills your own cells to prevent it. It's like a scorched earth tactic, right? Um, and then there's dendritic cells. Dendritic cells are also similar. They're white blood cells whose job is to go after pathogens that are inside you. So over here, when we are in our adaptive immune response, again, the adaptive immune response is when the innate immune response doesn't work. Dendritic cells take up the bacterial antigens at the infection site and travel through the bloodstream essentially. So this is actually the pathogen itself, or dendritic cell, my bad. They travel through the bloodstream to the lymph nodes, and native T cells circulating in the blood area enter the lymph nodes through they just entered the lymph node, my bad. Lymph nodes, my bad. And if they recognize the MHC complex, the T cell is activated by the dendritic cell and it proliferates for several days until it is a strong enough force to essentially fight the pathogenic infection. So, and it travels, again, everything travels through the bloodstream to actually figure everything else, figure everything out and deal with the disease. So, I'll explain the mechanism with the pictures. So here's the dendritic cell. Here's the site of the infection. It travels through the bloodstream over here to the lymph node. It picks up all these other guys here, and they travel back to the site of the disease, and that's where they deal with the disease. Now, this is this continuation of that. So after T cells are activated, memory B cells that were created during the innate immune response are activated by these cells over here. And when the antibody is bound by both the T cells and the B cell. Most B cells are only activated when they cross-link, which is when the B cell receptors cluster around an antigen, because several B cells are bound to repeated epitopes at the B cell receptor. So an epitope is essentially, again, I'll explain it later, but it's part of a pathogen. The memory B cells proliferate and generates plasma cell that secretes antibody against the antigen. So I hope everybody understood that, because this isn't you don't need to know it now, hopefully, but this is just an introduction to the immune system. It's crazy how the immune system works. So hopefully once this video goes up, you can come back to this as a reference. You can search up these terms to understand them better. All of these images were taken from mostly scientific papers, so you can probably reverse Google search them as well to figure them out. Now we get into the juicy part of all this. This is where we actually talk about vaccines in particular and how they work. So vaccines, something I came up with while working on this, they're kind of like a prequel to the immune response, right? Because the process of getting vaccine is essentially getting yourself sick on purpose. And that's kind of why I call it a prequel to the immune response, because you can't get the immune response without the vaccine or getting sick, literally. So now we straight up jump right into it, what different types of vaccines are. You guys probably heard live attenuated all the time. Now you actually get to know what it is. So the live, they essentially inject you with live but weakened pathogens. So they expose the pathogen to different chemicals or maybe radiation, not radiation, but usually chemicals that weaken it, and they inject it inside you. And it's meant to stay alive. It's not dead on purpose. Well, I'll explain why. It replicates on its own, and the dendritic cells, toll-like receptors bind with the antigens. And one example of this type of vaccine is the chickenpox vaccine. So you may be asking, why is it alive? What difference does it make if it's live or dead? The reason it's alive is because it's able to actually replicate on its own. That's the big deal. Now, I'll explain this in a bit more detail soon when we talk about adjuvants. Adjuvants are very important to know if we're going to talk about vaccines with people, but the reason these things are alive is because they're able to replicate on their own. And the reason it's important to replicate on their own is because that means that your body is going to deal with this a lot more seriously. Your body is going to see this and say, like, this is a threat we need to deal with as soon as possible. And it's going to have a much stronger immune response. And the stronger the immune response is, the stronger your memory B cells are going to be around. Because your memory B cells aren't there forever for every disease. Um, but if the disease is serious, again, like maybe this would be, your memory B cells are probably going to be there a lot longer because your body knows that this is a major threat to it. So, as we see here, this is from the CDC's website. Vaccine enters the cells and releases code to replicate itself. Yeah, and instructions. So another interesting thing, I didn't cover this. Um, this is for a virus. The chickenpox is a virus. The way viruses replicate is literally by using your own cells against you, right? They're not cells themselves. It's a debate whether viruses are even alive or not. But the way viruses replicate is, I don't know if chickenpox is a DNA or an RNA virus, but what they do is that they go to your organelles, they use your organelles and enzymes to replicate themselves. So, for example, if it's a DNA enzyme, DNA virus, they'd probably be using DNA primase. Or if it's an RNA enzyme, they'd be using RNA primase or DNA synthetase, my bad. And they get all this genetic material. They use your cell membranes to make a a capsule for themselves, and that's how they live. So actually, the reason why you have virus DNA in you from your ancestors is because that's what they do. Their whole purpose is to excrete their own genetic material into you, and that's how they replicate further. So as they're making more of themselves from your cells, eventually, what's going to happen is that here you go, we have this antigen presenting cell. The buddy comes up, takes in the virus, and he takes him over to do the process that we just covered for innate and adaptive immunity. So this creates again the memory B cells, and this is what leads to, yeah, immunity for chickenpox, for example, here. But there's other types of vaccines, by the way. So inactivated vaccines are different. These vaccines are just dead, right? They aren't alive at all. And I don't think with polio you want to take the risk of there being, a living pathogen in you. Um, Now, this works differently in the sense that they only have, whatever spike proteins or any other parts of the vaccine there or parts of the pathogen there. They don't enter any cells. The antigen presenting cell takes them just up immediately, right? Because they're not going into any cell. They're not able to go to your cell and inject their genetic material at all. They're just dead. That's all they are. They're often killed by radiation, heat, or chemicals. And this is the important part. Care is taken not to denature the proteins. I don't know how many people know how proteins work. I think we did learn this in high school and we went over this in college again. But like, let me just explain what it means by denature. Your proteins are wrapped in a certain way. So let's pretend my hands are protein. They're wrapped in a certain way. If the protein is in a pH which is too high, too low, temperature is too cold, too hot, it may not bend the same way. You know how, for example, if you take like a paper clip, it's bent in a specific way, and you put it in an area of a lot of heat, not enough to melt it straight up, but a lot of heat, it's going to bend differently because it's meant to be in a certain way, a certain room temperature for a certain purpose. And if you put it in an environment it's not able to be in, then that can really mess it up. Uh, that's one of the reasons actually why our body maintains a constant temperature because it doesn't want to mess up everything that we have going on inside of us. So why is it important not to denature the proteins though here? It's because the body is your immune system is going after specific proteins on the dead virus. Like for example, this thing has spike proteins. It's not necessarily what all viruses have, but this protein here is specifically a spike protein, like in COVID. If you are able to make these essentially the antigens that you go after, then um, you're able to deal with it when it comes for for real. So like if polio comes to you for real, then you already have knowledge of the spike proteins for polio. The reason why if they're denatured, it's a bad idea is because once they're denatured, they're different than usual. So like for example, let's say it's usually like this, it's denatured to this. Your antibodies are figuring out how to deal with this spike protein, not this one, which is the real polio spike protein. Again, I don't know if polio actually has spike proteins or not. I'm just giving this as an example. Um, Something important to note as well is that it's weaker than live vaccines because again, it's dead. It's not replicating on its own anymore. Um, so how do you deal with that? Because you need a bit of a strong immune response, especially for polio, such a dangerous disease like polio. You need a bit of a strong immune response to deal with everything. Now, it's actually similar in function to subunit vaccines. We'll get into that soon. But the way that they deal with this being so weak is that they use adjuvants. Now, adjuvants you've probably heard about without realizing it. But adjuvants are the chemicals that are used to make immune response more robust. The more robust the reaction is, means longer memory cells are stored. And it's actually one of the biggest reasons a lot of these vaccines hurt so much. I took the COVID-19 vaccine back in, was it 2020 or 2021? I actually don't remember. It's that long ago now. Um, I remember my arm, my left arm over here. The best way I can describe the pain I felt that night was I felt like somebody came with a baseball bat and just smashed every bit of my arm to pieces, like the bone to pieces. That's how much it hurt. I don't know why it hurt that bad, but part of the reason is adjuvants. Because the RNA vaccine, we'll get into that too. The RNA vaccine is not that robust. But the adjuvants there tell your body, hey, we're a really bad thing inside of you. Come get us. So uh, this is something that came into a lot of scrutiny during COVID-19. I actually had some friends who were very concerned about this. So they would use chemicals, they would put chemicals in your body, would be generally chemicals that you don't want inside of you. You don't want to put aluminum inside of you, for example. I think aluminum was one of the chemicals that they used, one of the metals that they use. They put all of these crazy things inside of you. And a lot of people are looking at this and thinking, I would never put this inside myself. And that's kind of the point, actually. The point is that you wouldn't put it inside yourself. The point is that you're calling attention to your immune system saying like, hey, this is a bad thing that's inside of me. And your immune system rushes in there and has a very, very harsh response against this. And actually, we have a little diagram here which actually shows the difference between a vaccine without adjuvants and a vaccine with adjuvants. So we have the immature antigen presenting cells becoming mature. They go to the lymph nodes and they have a pretty weak response here. So they just have the antibodies, B cells, and B memory cells. That's a pretty simple response. It should be expected that your body reacts this way. But with and you do have adjuvants, these little blue things here are meant to be the adjuvant. Let's say they're aluminum or whatever crazy material they put in there. Your body's going to send a lot more stuff to deal with it, right? Because there's a lot more crazy things in there. They're going to send the cytotoxic T cells. There's going to be a lot more antibodies, a lot more B cells, a lot of more memory B cells, and they're all going to be flooding that region to deal with this disease or disease because it's not really a disease right now. And that is the purpose behind adjuvants. Um, make sure you know that, make sure this is a very important term to know. Adjuvants are very important to know because a lot of the concern around vaccines comes around adjuvants because these are chemicals that you don't know that you're putting in your body. The point is that these are chemicals you don't want in your body because they make your body realize this is bad and they deal with it better. Now with subunit vaccines, they're part of okay, remember the word epitopes that we used before back when we were talking about the adaptive immune response? This is what an epitope is. They're essentially parts of the vaccine, which could be like a polysaccharide. Polysaccharide is literally just a fancy term for sugar or a protein. And that specific part of the antigen is put inside of you. So one example of this is actually the hepatitis B vaccine. That's how they do it. Again, obviously it does not replicate because you don't have anything to replicate with. You have even less than the dead vaccine actually. And that's why you have adjuvants in here. This little diagram here uses adjuvants to show us. Again, the mechanism, once you're actually in the body, you'll notice the mechanism is almost exactly the same for everything. So that's how subunit vaccines work. They're just epitopes. They're part of the vaccine. They're subunits, essentially, of the vaccine that are put inside you. And again, this is similar to the mRNA vaccine. And we're going to cover that a lot once we get to it. Um, Before we get to that, though, we have the toxoid vaccine. Sometimes it's not the pathogen or the pathogen which is bad for your body. Sometimes it's the side products of the pathogen. So there's a show. I forgot the name of the show. I can't remember. It's about one of the ships, one of the explorer ships from Britain that went up to the North Pole or went to Canada to do some exploration work. Um, what was the name of the disease? There was a disease that they got. If somebody's able to put in the chat, it's essentially that disease you get from canned food because the spores are still in there. Something similar to that can occur here, where it's not even where sometimes you have to deal with the spores. Actually, honestly, I think that's more subunit vaccines than toxoid vaccines. Um, I should give a better example. My bad. But diphtheria is actually a disease that is caused by this, where it's not necessarily the bacteria that's bad for you, but the side products of the bacteria. Now, a toxoid vaccine is essentially a vaccine made to deal with the toxins, right? So they use toxoids, which are inactivated toxins. Sometimes these can be proteins, thus you heat kill them, etc. And they put them inside your body, and once your body actually, so this is actually the most different of the vaccines. Once your body deals with them or it comes into contact with them, it makes the antibodies that deal with it. The antibodies bind to the cell. And as a result, when the real antibody or when the real toxins come in, your antibodies know exactly what you need to do. Now, for this vaccine in particular, by the way, you may need multiple doses just because of the fact that the antibody levels do fade over a period of time. But you'd probably prefer this over getting the real toxins. Remember, toxins are anything that can cause damage to cells, damage to your body. And as a result, a toxoid vaccine is really helpful in that regard. Now we get to the real spicy stuff, the mRNA vaccine. You're injecting people with genetic material. This is like the big scary thing. This is what COVID vaccine is actually. So we have something that kind of looks like COVID over here. Um, and this was always like something scary. I know so many people come up to me saying like, oh, they might change your DNA. So before we even get to the mRNA vaccine, we need to understand what mRNA is and how it works. So DNA, if you guys know the central dogma of biology, it's DNA is transcribed into RNA, which is translated into, wait, hold on, I'm saying it wrong. I put it wrong here. My bad. No, wait, I am saying it right. My bad. DNA is transcribed to RNA, which is translated into proteins. You can remember it this way. Because DNA and RNA, they kind of use the same language. They're both still adenine, cytosine, thymine, guanine, except uracil is thrown in there. So it's like a slightly different, it's like slightly different dialect, of a language. So you're essentially writing from one to another. You're using the same alphabet, right? So if you're an English speaker and a Spanish speaker, since they use the same alphabet, you can kind of figure things out with cognates and stuff. It's just transcribing things, right? But when it's RNA to proteins, RNA and proteins are not related at all. I mean, they technically are, but they look nothing alike. And as a result, it's completely new translation of one language to another. That's one way you guys can remember it. That's how I remember it. So DNA is transcribed into mRNA by enzymes, right? We're not going to go into the nitty-gritty of that. That's all biochemistry stuff. But if you guys want to look into that, these are the terminologies to search up. The mRNA matches with a certain tRNA at a ribosome to produce an amino acid. So this is how it works. I'll explain it now. So we have the mRNA right here. This is the going from the 3' to 5' direction. This is the opposite of the DNA. So the DNA here would be 5' to 3'. We have uracil here. The opposite would probably be a DNA, which is A, C, C, T, T, T. Okay, I'm going too much into detail there. But each one of these three mRNA codes is a codon. That's one way to remember it, a codon, right? They code for something. And when they get into the ribosome, which has a large unit, a small unit, these codons have to match with anticodons in the tRNA. The tRNA, by the way, stands for transport RNA, mRNA messenger RNA. So the mRNA, the messenger RNA, gives a message to the transfer RNA, which protein it needs to match with. So they bind together on the opposite in the ribosome. So for example, if you have AAA, if you know, or adenine, 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 the opposite of adenine in RNA would be uracil. So uracil, uracil, uracil binds to this one. And then this one, guanine binds to cytosine, adenine to uracil, and uracil to adenine. And each one of these codons codes for one protein, one amino acid. That's a better way to put it. And amino acids are basically the building blocks of proteins. And every time, so actually this diagram on the left explains this a little better than this one. So what happens, the actual mechanism is this guy will come in, he'll have his bond to his little codon here. The codon shifts to the middle, and his amino acid is shoved upwards into this amino acid chain, which is made by peptide bonds. And finally, he shifts over when this guy comes in, and it's his turn to do it, and he leaves. Meanwhile, his amino acid, which was just removed, it is moving into a chain little by little. So actually over here, we see the growing peptide chain. We see more detail, the actual way that the messenger RNA interacts with everything. So note that the important takeaway here is mRNA does not change your DNA at all. Your DNA is completely untouched by mRNA. The mRNA still uses your enzymes, it uses your ribosome to code for proteins, but it does not touch your genetic material. Now, to explain why proteins are important, remember the amino acid is the building block of protein. Once the amino acid chain becomes long enough, actually, so I'll explain with the little hand thing again. So my hand is like this, right? This is essentially like my fingers, they're the amino acid chain, but they wrap around each other in a specific manner, which is how the protein is. So this my hand is a protein. When they're denatured, it's because something occurred and caused it to fold wrong. So now that we understand how mRNA actually works physically, we can move over to the mRNA vaccine. So the mRNA vaccine uses host cells to generate proteins that become antigens essentially. So it's like the spike proteins in SARS-CoV-2 or COVID-19. So in this little diagram here, I don't know how easy it is for you guys to see. These little blue things around the red and yellow, those are the spike proteins of the SARS-CoV-2 virus. Now, the way that mRNA vaccines work is that they only replicate once, and it is then targeted by the immune system. So actually, again, if you look here, these are just going in once, right? Once the mRNA is done, the mRNA is done, right? The mRNA is actually disposed of by the body once it's finished. Because the mRNA's job in your D I'm going in a full nerd mode right now, but I need to explain this. When you have like, let's say this is a whole chromosome, right? But it's a pretty small chromosome, honestly, but like, let's say this is part of the chromosome, right? Part of your DNA. The mRNA only comes from this little like bone right here of my finger. This little gene is all your body cares about, right? So it makes a complementary mRNA. So let's say this part of my finger right here, my other finger, is the mRNA. This goes into the ribosome and produces the actual protein. Once this thing is done, it's not neat anymore. It just becomes junk that your body doesn't need because your body is very efficient. It only keeps the mRNA or proteins that it needs to work. Once that is done, the mRNA is gone. So this mRNA right here, gone, done. That's it. So it only replicates once. So you probably want to have quite a bit of mRNA injected inside you once. But the mRNA being there alone is not enough. Adjuvants are also used in mRNA vaccines. That's actually why they hurt so much. And that might be what causes some of the side effects, like the expanded heart and whatnot, like the swelling of the heart, the carditis. That's not necessarily the result of the mRNA itself. That's likely the results of the adjuvants because the mRNA is just mRNA. It's not going to do anything crazy different that your body didn't already do. So here are some concerns during the COVID pandemic about it. One concern was changing of DNA. Again, as we explained, once your mRNA is done, or mRNA is done, this is the finger for it. Once your mRNA is done, it's gone. That's it. It doesn't go back into the DNA. It doesn't go anywhere else. Your body gets rid of it, or your cells get rid of that. So your DNA remains unchanged. You're more likely to actually have your DNA changed by the prior examples of the weakened pathogens, because the weakened pathogens are still alive, so they're still able to inject their DNA or RNA into you. So that's much more likely to happen. I just realized I'm kind of getting a bit late on time, so let me try to get through this fast. Unknown substances of vaccine, again, adjuvants, we know about them. Now, sudden deaths were also a major concern. Just keep in mind, people have always died suddenly. It's sad. It's a reality of life. It's possible that I can fall dead on my screen right now. It's not necessarily something to do with uh, the vaccine in particular. So let me just go into a few more things. A lot of people have concerns about vaccines don't work. No medication will have a 100% success rate. So many factors take part in whether treatment succeeds or not. It could be health. It could be, it could be anything. It could just be random chance. Maybe that specific vaccine was a dud. During COVID, patients who took the vaccine and became symptomatic, again, were shown to still do better than those who did not take the vaccine. So less deaths were noticed among these populations in hospitals. Um, Note as well, mutations are found in all living things. Even in us, we're mutating by accident. Our bodies are probably killing cells right now that mutated the wrong way. Mutations are found in all living things, even us. And that's why there's a new flu vaccine every year. It's not some conspiracy to, make you take the vaccine more. I don't care enough about the flu vaccine to take it all the time. But the flu vaccine is an example of diseases change. COVID's going to change. COVID's going to mutate. It's fine. It's just a reality of life. The reason that they're giving more doses is just so that you're able to deal with it stronger. And note that at the time, the reason the vaccines were pushed so much was that COVID right now is not as deadly as it used to be. I remember looking at the death charts of COVID. It was insane how bad COVID was at the time. But right now, you barely hear about COVID. People get COVID. I think my roommate got COVID over winter break, and he doesn't care. He just got COVID. He dealt with it, and he's done. Right? But like back then, if you got COVID, it was a very severe thing. It's a new disease. People were concerned, and on top of it, it was a lot more deadly at the time. So note that the reason what vaccines were pushed to begin with at the time was because of the concern. It wasn't just because they wanted you to get vaccinated for no reason. Right. Again, sudden deaths, they came under scrutiny after COVID. As I mentioned, it can happen all the time. It's not necessarily because of vaccines. We know there were severe side effects, but this is not hidden about it. The reason why a lot of people look at it more is because there's intentional bias. You're looking at this a lot more now But sudden deaths have always been a thing. Again, our lives are not in our hands. Our lives can end at any moment. As I said before, I could have a sudden heart attack while talking to you guys right now. It's the reality of life. And always remember, correlation does not equate to causation. Because somebody got the vaccine and then ended up getting a heart attack, it doesn't mean the vaccine caused a heart attack. It could be a number of factors that could have caused it. It could be anything. So always keep that in mind. Now, the final point we have, we're at 10 o'clock now. Let's try to get this done fast, is hesitancy. Explain vaccine hesitancy. This is a very important thing. I'm going to try my best to explain it without rushing, but I'll do my best. So 22% of Americans express sympathy with the anti-vax movement. This is a very large number of Americans. That's one out of every five Americans, more than one out of every five Americans. And I have some friends who have expressed sympathy with the movement. And across political lines, Republicans, obviously, actually, this chart's actually kind of crazy. 2016, 2019, everybody kind of was okay with vaccines. But then by March 2023, this is not even a year ago, almost a year ago, we saw that Republicans did not trust vaccines at all. All across all US adults, there's a general lack of trust in vaccines. So this is the whole situation right now. Vaccine hesitancy has increased greatly over the last few years. Again, 42% of Republicans are hesitant about vaccine. So how do we deal with this? We have to know how to address the concerns. We have to understand the concerns are very legitimate. These are four images that I picked of the lies or atrocities or whatever you want to say of the US government. African Americans are shown to have a lot of hesitancy when it comes to vaccines. Part of this is because of the Tuskegee experiments. African Americans were injected with syphilis as tests to see, how do African Americans get affected by syphilis. And eventually a cure was developed, but these people would never receive the cure. They just got syphilis. They got paid, they got food and stuff, but they never got the cure for syphilis despite being tested to see how syphilis works. And as a result, a lot and African Americans, this wasn't just this. There was whole Jim Crow, there's slavery, there's all these terrible, terrible things that happened to the African American community. Um, there's a historian on YouTube named The Cynical Historian. That's a YouTube channel name. I don't know his actual name, if I'm being honest. I watched him for a couple of years. He said that America is actually an anomaly when it comes to most societies. Like this doesn't happen in Rome. Rome had a lot of slaves too, but only in the United States can you look at somebody and have an idea of them potentially being like an ancestor or a descendant of a slave. That doesn't exist in any other society. There's no other society where you just look at somebody and know that, oh, their grandfather was probably a slave. So the United States is unique in that regard because of this. So the African American community has a lot of genuine mistrust of our institutions. We also saw that a lot of people hesitant about vaccines were white. And again, a lot of white Americans were lied to too. This is an image here of the taking down of Saddam Hussein's statue in Baghdad during the 2003 invasion. Our government told the American people and the American military, overwhelmingly white, that we were going in there to remove WMDs, and Saddam had these WMDs, and he wanted to use them against us, all this sort of stuff. And then eventually, everybody found out that no, this wasn't true at all, and we lost 3,000 Americans in Iraq for nothing. And then even more with PTSD and military and suicide from that. So this actually shattered white Americans' trust in the government completely. And it's a legitimate reason why, because your sons and daughters are going to this country to fight for essentially nothing. Native Americans also do have a bit of concerns about the vaccine. Why? This is an image of the Trail of Tears when I believe it was President Andrew Jackson forcefully removed Native Americans from the South and moved them out to the West. A lot of their land was taken, a lot of it due to violations of treaties. They didn't even get genuine grievances or addressing of concerns until maybe the 70s. So Native Americans also faced a lot. And another example is just to show that even communities which are well off today weren't always safe. These are pictures of the Japanese internment camps during World War II, where Japanese Americans in the West Coast were rounded up and sent to these camps because people were afraid of them being, Japanese sympathizers. So understand that. People are scared for a good reason because things have been done in the past by our institutions that we're supposed to trust, right? There's a reason why these institutions don't have trust right now. So what do we do about this? Our job is to explain the science, and we have to be humble. Why? Because when you explain the science, only then will you actually shift perceptions of vaccines. People aren't going to trust the CDC. The CDC is literally a government body. Nobody's going to trust the government anymore. And honestly, they have a point. Again, I think we should also ask questions about why is the government doing this? Not necessarily to say that the government is wrong, but to understand so that we can help others understand. And we should be humble. We should recognize, we should get on the same level as people. We should recognize that these are people who know what our institutions did in the past. If we're trusting them blindly, we actually don't know what our institutions have done in the past. So get on their level and explain the science in a humble manner. And here's some tips that have worked with me to making people a little less hesitant about vaccines. So explain what you know about how things work with them, right? This is like the most simple thing you can do. Explain how what you know to them and explain like the mechanism behind vaccines. Don't say like, "Oh, Dr. Fauci said this. It's right. He's a doctor." Never use that argument. There's a reason they don't trust Dr. Fauci. It's because he's in the government, right? It's and this is not a disrespect to Dr. Fauci. It's just the fact that people don't trust him because of his position, because of past misgrievances. So explain what Dr. Fauci is saying to them with like the science you know, and give them access to scientific papers, and tell them that they can email the people who are in scientific disciplines to have these explained. They can email even the writers of these scientific papers to explain what this means to them. So, or you can even explain it to them if you understand it yourself. Let them know that the scientific field is one of the most transparent and scrutinized fields in the world. I mean, just the fact that those two studies I mentioned before were so heavily scrutinized, this is how science works. And explain the scientific method. A lot of the reason why COVID was so weird to a lot of people was this is how my microbiology professor, Dr. McAllister, Nicole McAllister here at Seton Hill explained it to me. They were essentially watching science work in real time. They're watching us discover new things in real time, watching new hypotheses and new theories come up in real time. So this is the scientific method. The reason why all these contradicting things came up was because we were learning new things in real time. The reason we know about let's say the reason we know about gravity, the way gravity or black holes is because we've had so much time to refine black holes, is because this is something that was going on and off the whole time. Einstein did math discover the black hole, and eventually more people supported that math, and then and then eventually it was photographed. Um, that's how science worked. Not everybody agreed with the black hole to begin with. Not everybody agreed the Earth was round to begin. Not everybody agreed that the Earth orbited the sun to begin with. So all these things take time. It was just that we're watching science in real time as a society as a whole. Usually, science, society as a whole, unless people are interested in it, just ignore how science works in real time. But we're watching this in real time. That's why things were changing. So explain the scientific method and make sure the information is easy to digest. So I tried my best to do that. I hope you guys understood me with like the whole thing with my fingers making different things. Make sure it's easy to digest so that people can understand. Do not be technical because people need to understand things in the mainstream. We're in college for how many years? We have all the scientific jargon. It's easy for us to understand, but we have to give it to the general population. We're here to serve the general population of this world, not ourselves and not other people in our places. So that concludes the hold on. I'm having some trouble right now. How do I open this? Okay, there you go. All right, so that would be the end of the session for now. The link to the quiz for the session is now live and it's being sent in the chat box. Um, I hope to see you all for our next session next Saturday, February 10th at 9 a.m. Eastern Standard Time. And this concludes this week's shadowing session.